Hi, I'm Joe Ruhl, and I want to welcome you to this lecture demonstration on DNA, RNA, and protein synthesis. We're going to be looking at this wonderful molecule, this DNA molecule that is found in the cells of all living things on the planet. In fact, this DNA could have come from a mushroom, it could have come from a bacterium, it could have come from a corn plant or a mouse or your dog or your cat or a monkey or you. It's all the same stuff in all living things on the planet. It's a pretty impressive molecule. Now, what we're going to do today is we're going to look at how it's put together and who discovered how it's put together. And then we're also going to look at how DNA is able to make copies of itself, which you'll find out is very important. And then finally, we'll look at how DNA, which contains a recipe for you, is able to control the manufacture of all the protein molecules that you're made of. And so in a sense, that's how DNA controls what you look like. It controls the manufacture of the protein molecules that you're made of. And we'll take a look at how all of that works. Now, first of all, I would imagine you're probably already familiar with molecules. For example, let's look at a simple molecule that is so vitally important for life on Earth. This water molecule. And you probably already know that it kind of looks like an upside down Mickey Mouse schematic. It's made of 2 hydrogen atoms bonded together with 1 oxygen atom. So it's just 3 atoms bonded together to make this very simple yet very important molecule. Now DNA, on the other hand, if we look at this DNA molecule here, is actually in a class of molecules called macromolecules. And macro means big. Actually, it's kind of an understatement because this is a huge molecule. We're only looking at a small length or piece of DNA here, and we're going to find out as we go that it's a huge, huge molecule. So sometimes you'll hear about chromosomes and genes and DNA. You'll hear about those words being used maybe interchangeably, and maybe you wake up in the middle of the night wondering, how are chromosomes and genes and DNA related to each other? Well, take a look at this picture, and I think it'll give you a good idea. You may be familiar with chromosomes. And so here in this picture, we've got a picture of a chromosome on the left, and you may realize that we humans, most of us humans, have 46 chromosomes, kind of look like this. And if you imagine unraveling a chromosome, grabbing the tip of it and unraveling it and stretching it out, you'll see that it's actually a long, coiled up DNA molecule. And again, DNA looks a little bit like a ladder. In fact, we're going to see it looks like a twisted ladder or maybe a spiral staircase. But each chromosome, and you've got 46 of them in every cell in your body, each chromosome is a long DNA molecule that's all coiled up. So there's the connection, there's the relationship between DNA and chromosomes. Now, what are genes and where are they? Look at this little segment here of DNA. That might be a gene. So for our purposes, we're going to say that a gene is a small length of DNA that contains instructions in it or a recipe for how to make a particular protein molecule. So there's the connection, you see, between chromosomes and genes and DNA. Now, how many genes do we have in these 46 chromosomes? It's a little over 20,000 gene pairs. So that's a lot of information stored inside those 46 chromosomes inside that tiny nucleus. Now, I want us to gain some appreciation for not only the beauty of this molecule, but the small size of it. Let's take a cell, an egg cell, human egg cell. Now actually, it's the largest cell in the human body. The diameter of a human egg cell is about the diameter of, it's about the width of a hair. And so another way to look at it is if you took a really super sharp pencil, as sharp as you could get it, and lightly touch it to a piece of paper and make a dot that you can barely see, that people under 35 might be able to see with the naked eye, that's about how big a human egg cell is. It's the largest cell in the human body. Now, imagine if we enlarge a human egg cell to, let's say, this size. The nucleus inside would be about the size of a softball. If it's a fertilized egg, if it's joined up with a sperm cell, which is like millions of times smaller than the egg, if it's a fertilized egg, there will be 46 chromosomes inside that nucleus. 23 from mom, 23 from dad. So we're talking really tiny here. And those chromosomes, remember, are made of this molecule DNA. So we're talking about stuff that's really small here. Now, how do we know DNA has this three-dimensional structure? Kind of looks like a spiral staircase or a twisted ladder. How do we know that? Well, take a look at these two men in this photograph, James Watson and Francis Crick. Way back in 1953, that's 2 years before I was born, they discovered through lots and lots of painstaking research, hard work, they discovered the three-dimensional structure of DNA, 1953. Actually, they didn't do it alone. Scientists hardly ever work alone. They collaborated with this gentleman right here, Maurice Wilkins. And together, there were other people involved. But Watson and Crick seem to get most of the credit for it. In fact, in 1962, James Watson, Francis Crick, Maurice Wilkins won the Nobel Prize for discovering and publishing the three-dimensional structure of this molecule. There was another person who was involved who doesn't get a lot of credit. In fact, it's not fair, but she did not win a share of the Nobel Prize, but her work was vitally important to the discovery of the three-dimensional structure of DNA. Her name was Rosalind Franklin, and here you can see a picture of her. And she was a brilliant scientist and did some work in an area called X-ray crystallography. And she produced this photograph. This is a very famous photograph. It's called Photo 51 of the structure of DNA. Watson and Crick had been struggling, trying to figure out the three-dimensional structure. They were missing a vital piece of the puzzle. And wouldn't you know, Rosalind Franklin was the one who came up with the missing piece of the puzzle that they needed to complete their work. The key was this photograph right here, Photo 51. Sadly, she died at a young age in 1958, and at the age of 38. And so she did not receive a portion of the Nobel Prize in 1962 because you cannot win the Nobel Prize after you have passed away. So she was ignored for a long, long time, but now she's getting her rightful credit, and I'm happy for that. I wish she would have been around long enough to realize just how important her work was. So what did Watson and Crick and Wilkins and Franklin discover? Well, you're looking at it right here. Three-dimensional structure of DNA. Now, what I'd like to do is take that DNA molecule, simplify it a bit, and here it is, simplified. It's a ladder. So imagine I've taken the ladder and I've just untwisted it and I've laid it out flat on the board right here. So this is our DNA molecule. I want to look at the parts of it. And so we're going to start with this molecule up here. Now let me just take this off here. This block labeled P, that's actually a phosphate molecule. The block labeled S, S is for sugar. That's a sugar molecule. Now it's not like the sugar that you might have on your kitchen table in a little container. That sugar is called sucrose. Oh, and it's not glucose, that's the sugar that plants make when they do photosynthesis. It's a sugar molecule called, in this case, deoxyribose. And so the molecule's name, the whole molecule's name, deoxyribonucleic acid, comes from the fact that the sugar in it is called deoxyribose. This letter, this block attached to the sugar molecule, labeled A, it's one of 4 different kinds of, we call them bases. And we'll look at those in detail as we go. But I just wanted to point out to you the main parts of the DNA molecule. And you'll notice if you look closely at this molecule, the steps in the ladder are formed by these bases, these molecules called bases that are joined together. 4 different bases, T and A and C and G. Now, you're probably going to notice a pattern right there. T always pairs with A, C always pairs with G. It's almost like C and G like each other, and A and T like each other. They're attracted to each other. Sometimes when I'm in the hallway watching students during passing period, I can see A's and T's being attracted to one another. Well, students playing the roles of A's and T's. They may not realize they're doing that. And other students, C's and G's, being attracted to one another. I see that all the time. Here we have students acting like DNA bases and they don't even realize it. Happens all the time. So there are some patterns that you can see in this DNA molecule right here. Now, we normally don't call these steps in the ladder No, scientists, when they're talking about DNA, they refer to each step as a base pair. So these steps are called base pairs. How big is this DNA molecule on the board right here? It is 1, 2, 3, 4, 5, 6 base pairs long. That's how big that DNA molecule is. 6 base pairs. Now, if we look at our little more complicated, more realistic model of DNA, I don't know if you can see these or not, but I'm going to count them for you. There's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16 base pairs in this DNA molecule. So, in just one of your cells, and you're made up of like 100 trillion cells, in just one of your cells, in those 46 chromosomes, how many base pairs of DNA do you think are in those 46 chromosomes? Well, I'll tell you the answer. It's an estimate. How many base pairs? A little over 3 billion base pairs. Not 6 like this, not 16 like this, 3 billion. Now, that's a number that's almost impossible for us to imagine. At least it's impossible for me to imagine. So I like to build models to try to understand, like, what is 3 billion? I mean, how big is it? And here's something that'll help us to understand that. If you enlarged your DNA to the size of this model right here, and make it not just 16 base pairs in length, but 3 billion, if you made it this big, 3 billion, how long would that 3 billion base pairs worth of DNA be? If it was this size, it'd stretch around the world around a dozen times. Do you see what I mean when I say macromolecule? Macro, that's kind of an understatement. This is an amazing molecule. It's huge. Here's another way to look at that. Look at these base pairs here. TA, AT, CG, GC, AT, CG. Now, if you were to try to read all 3 billion base pairs of your DNA, like this fast, TA, AT, CG, GC, AT, CG. How long would it take you to read all 3 billion base pairs? The estimates are, if you did that out loud, read them out loud, 7 days a week, 24 hours a day, nonstop. It would take you 31 years to read off all 3 billion base pairs. Here's a diagram just like the one on the second page of your handout that shows the parts of the DNA molecule that we just looked at. Notice the actual names of the 4 bases: adenine, thymine, guanine, and cytosine. But we're just going to refer to them from here on out as A, T, C, and G. Now, there's also beauty in nature if you look hard enough, if you know where to look. If you look like a cross-section, look at a cross-section of this DNA molecule. In other words, imagine we're looking down the barrel of this DNA molecule. It'd be sort of like standing at the top of a spiral staircase and looking down. But here's a photograph. It's actually generated or produced by a computer of that kind of view, looking down the barrel of a DNA molecule, a cross-section. Each little colored dot in this photograph is an atom, represents an atom. So look at the beauty in nature if you look hard enough and if you know where to look. It's a beautiful molecule, it's elegant, and it works so efficiently. If we go back to this model here, again, a similar kind of arrangement here. Each of these colored balls is an atom. So, as I said, macro in the macromolecule, kind of an understatement. Now, the neat thing about DNA is it's able to make copies of itself. DNA is able to replicate itself. And that process, of course, as you might imagine, is called DNA replication. And I want us to look at how that works. It's a simple model here. It's a little more complex than I'm going to show you, but I think you'll get the basic idea of how DNA is able to make copies of itself. Now, why is it important? Imagine, and maybe you've learned about cell division. You started out life as a single cell, a fertilized egg, which went through cell division to become a 2-celled embryo, and then those cells divided again to become a 4-celled embryo, and then 8, and then 16, and 32, and so on. Now look at you, you're 100 trillion cells. Every time a cell divides to make 2 cells, it has to replicate all of its DNA, so that each of the 2 new cells will have a complete set of 46 chromosomes made up of 3 billion base pairs of DNA. And that process has to go without a hitch. In fact, it's amazing when DNA replicates. It only makes a mistake, like 1 out of every 100 million bases when it replicates. Each mistake when DNA replicates is called a mutation. And we're going to look at that in a little more detail as we go. Wow, 1 base change, 1 mistake out of every 100 million letters. I'll bet you can't type a paper with 100 million letters in it without making a mistake. I know I couldn't. DNA does it most of the time without a hitch. Let's look at how DNA replication happens. First thing that happens is the 2 sides of the DNA molecule split apart, and we call that unzipping. They unzip. Now, I don't have enough hands to show you, but the bonds holding these building blocks of DNA together, for example, these 2 building blocks of DNA, the bond holding them together, those are very strong chemical bonds, and so the whole side of the ladder doesn't break apart like I'm going to show you, and it's simply because I don't have enough hands. But these basic building blocks of DNA right here are called nucleotides. So what I'm holding in my hand right here is a nucleotide. So I'm going to put that right back here. So let's show DNA starting the process of DNA replication. Let's show it unzipping, which is like the first step. So here we go. We're going to unzip. We're going to separate the two halves of the DNA molecule like this. And like I said, these nucleotides don't break apart. They stay together, kind of like that. All right? So now the two sides of the DNA have unzipped. Now, there are going to be a whole bunch of other nucleotides, free nucleotides, just floating around in the nucleus of the cell here. Pretend that this chalkboard now represents the nucleus of the cell. And in here we've got all these free, unattached, lonely nucleotides floating around, and they would love to be paired up with their partners. So I'm going to put a bunch of these free, unattached nucleotides in the nucleus of the cell here. Okay, here they are, a bunch of them. Now, by the way, DNA doesn't unzip by itself. There are other molecules in the cell called enzymes, and those are special protein molecules that do work. And these enzymes actually do the work of unzipping the DNA. There are special enzymes that do that. So my hands maybe here would represent those enzymes. Have you ever bolted up in the middle of the night and wondered, where do these free nucleotides in the nucleus of the cell, where do they come from? Where do they come from? Well, there are other chemical reactions going on in the cell that actually manufacture these nucleotides from raw materials. Now, where do you get the raw materials to make nucleotides? Well, they could come from the carrot or the lettuce or the chicken or the cow that you ate yesterday. Because see, when you eat muscle tissue or plant tissue, muscle tissue, that's what meat is, from other animals, or plant tissue, those living things have DNA, and so your digestive system takes the DNA from those living things that you eat, breaks it down into nucleotides and other building blocks of nucleotides that are delivered from your digestive system by way of your bloodstream to all the cells in your body. That's where they come from. So you are what you eat. Really. All right, now let's watch DNA replicating. Remember I said T and A's are attracted to each other and C's and G's are attracted to each other. And so you get this pairing up going on now, aided by enzymes. This lonely A is probably going to be attracted to this lonely T right here. And at the same time, maybe this lonely C is attracted over here to this lonely G. Don't fall head over heels in love. And then maybe this lonely T here is attracted to this lonely A, and this lonely A attracted to this lonely T here. And this G may be attracted to that C. What you're watching here is DNA replication. And this G might be attracted to this C, and this C to this G, and this C to this G, and maybe this T to this A, maybe this A to this T, maybe this G to this C, maybe this T to this A. All right, we've just witnessed DNA replication. And what do you notice? If you compare these two DNA molecules to each other, what do you notice? Well, I hope you notice that they're identical. They contain identical instructions. The order of these base pairs spells out coded instructions, part of the recipe for you. This might be part of the recipe for how to make toenails, or an enzyme in your stomach that breaks your food down. Or in another living thing, this might be part of the recipe for how to make a protein called rattlesnake venom. So that's why the order, the specific order of these base pairs is important. Now, sometimes when DNA is replicating, it makes a mistake, or the enzymes responsible for replicating the DNA might make a mistake. Like, for example, what if when DNA is replicating, the wrong base is put in, let's say, here? That doesn't work very well. They don't fit very well. But sometimes the enzymes make a mistake when they're replicating DNA. Now, fortunately, there are other enzymes in the nucleus of the cell called proofreading enzymes that are able to detect that and correct it and cut it out and put the correct nucleotide in there. But sometimes the proofreading enzymes will overlook the mistake. Have you ever done a spell check when you're typing a paper on a computer and you type a word that's incorrect, but the spell checker doesn't catch it? Like maybe T-H-E-R-E, where it should say T-H-E-I-R, or something like that. Sometimes proofreading enzymes won't catch the mistakes. And if the mistake stays in there, like that, then it's passed on faithfully, generation after generation. Every time the DNA replicates from that point on, the mistake is passed on, and this change is called a mutation. And lots of times, these single base changes don't cause any problems. Sometimes they do cause problems. Sometimes they're even lethal to the organism. And in some cases, they may even make the organism better off, a little bit better able to survive than its peers in its particular environment. So that's kind of the basis of DNA replication. And like I said, why is it important? Every time a cell divides in two to make two new cells, it has to faithfully replicate all of its DNA so that the two new cells each get their fair share of the correct amount of DNA. Now, I want us to, again, gain even more appreciation for the complexity of this molecule. Take a look at the sugar molecule, deoxyribose. It doesn't really look like this. If you look at this next picture, this is a little bit better picture of what that sugar molecule, deoxyribose, looks like. And you can kind of see that it's made up of a bunch of carbon and hydrogen and oxygen atoms. So I want you to gain, like I said, appreciation for the complexity of this molecule. We don't have to memorize the structure of it, but just have some appreciation. Show some respect. And the bases, A and T and C and G. Look at this next picture. This shows what these four bases actually, a little bit more realistically, look like. And you can see that they themselves are pretty complex molecules. So DNA is a wonderfully complex molecule. What I want to look at next is the process of how your DNA controls the manufacture of you. How DNA controls what you look like, how DNA controls the manufacture of the protein molecules and the enzymes that you're made of. And that process is called protein synthesis. So that's what we're going to look at next. So we just looked at how DNA is able to replicate, make copies of itself. The only time DNA does that is before cell division, as we talked about before. But what's DNA doing all the rest of the time when the cells are not dividing, when the cells are just doing what cells do, when liver cells are just being liver cells and pancreas cells are just doing what pancreas cells do, and your brain cells are just doing what brain cells do. Everyday activities. when they're not actively dividing and undergoing cell division. So what's your DNA doing? Well, it's controlling the manufacture of the protein molecules that you're made of. This process is called protein synthesis. Now, it can be summarized with this diagram right here. And this idea in biology sometimes is referred to as the central dogma. In other words, it's like the main idea of biology. It's what all of biology is based upon. In fact, all living things do what you're looking at in this picture. Slime molds, mushrooms, bacteria, yeast cells, plant cells of all kinds, animal cells of all kinds, dogs and cats and rats and elephants and people, they all do this. And the process looks something like this, and this is like a simplified diagram of it. It reads like this. DNA makes a molecule called mRNA, or messenger RNA. Messenger RNA then gets translated, the instructions in the messenger RNA are translated to build a protein molecule. And as you can see in this diagram, proteins are equivalent to traits, or you might say your traits are protein molecules or collections of protein molecules. And so this process, the central dogma, is vitally important. And like I said, all living things do it. And I want to look at that process right here. Now, if we come back here to the chalkboard, look at our DNA molecule. Again, this is just a tiny little piece of DNA. Remember I said all the DNA in just one of your cells is like 3 billion base pairs, not 6 base pairs like we're looking at right here, but 3 billion. If you typed all that out, it's something to think about. If you typed out all 3 billion base pairs, the recipe for you, how to build you from scratch, type those letters out. T, A, C, G, A, C, for example. All 3 billion. If you type them out, let's say you use Times New Roman font, 12-point size, not bold, but regular font style. You type them out on paper, single-spaced. No margins, top or bottom of the paper, or left or right. Totally filled the paper with bases. T's and A's and C's and G's. No spaces between the bases. No spaces between the bases. No margins on the paper. Print those on both sides of the paper. How big would that recipe for you be? It would be a stack of those papers. I kid you not, as tall as the Washington Monument, like you can see in this photograph right here. That's how big the recipe is. And that entire recipe is referred to as your genome. Genome, or the human genome. And all living things have their own genome. My dog has a genome that's made up of base pairs, A's and T's and C's and G's that spell out instructions for how to be a dog. It's found in all living things. No wonder it's called the universal molecule of heredity. But what I want to do right now is I want to look at how DNA is able to control the manufacture of the protein molecules that you're made of. Now, first of all, DNA doesn't do it all by itself. There are 3 other kinds of molecules that work with DNA. They're a little bit like scientists. They work collaboratively. Most scientists don't work all by themselves. They work with a team. Same thing with enzymes and molecules in your body. So there are 3 kinds of molecules we're going to look at that help, you might say, DNA. And if we go back to the central dogma diagram, you'll notice it says DNA makes RNA, RNA makes proteins. There are 3 different kinds of RNA molecules involved here. First of all, we have one that's called messenger RNA, or we abbreviate that mRNA. And then there's another kind called transfer RNA, and we call that, or we abbreviate that, tRNA. And then there's a third kind called rRNA, and that stands for ribosomal RNA. Now, I hope you remember when you studied the parts of a cell, I hope you remembered what ribosomes are. Take a look at this picture of a cell. Notice the cell parts that maybe you can remember some of those. I hope you do. Notice the tiny little dots that are pointed to in this diagram. Those are the ribosomes, probably the smallest, among the smallest of the structures inside of the cell, but they're so important because these little ribosomes are kind of like the workers in this factory that we call a cell, and these ribosomes build protein molecules. That's what they do. Now, how do these ribosomes build protein molecules? It's a little bit like this. Protein molecules are made of smaller molecules called amino acids. I happen to have an amino acid molecule right here. And here's another kind of amino acid molecule. It's a different kind of amino acid. And here's what ribosomes do. Pretend like I'm a ribosome. All day long, this is what ribosomes do. They take amino acid molecules and they string them together like this to build proteins. So here I am, combining together these amino acid molecules, stringing them together to build a long string of amino acid molecules called a protein. There it is. Now that's a pretty small protein molecule. That's just a model. But the idea that I want us to keep in mind is that a protein molecule is a long chain of amino acids. There are so many different kinds of protein molecules in your body that have to be manufactured following the instructions in the DNA. All kinds of protein molecules. For example, hemoglobin, the molecule in your blood that carries oxygen. It's actually 4 pretty big protein molecules joined together to make a big, what we call hemoglobin. That's an important molecule. All the enzymes in your body that run the chemical machinery, all the chemical reactions that happen in your cells, all those enzymes, all those different kinds of enzymes, they're protein molecules. The building materials that make up the structural components of the cell, those are protein molecules. So without proteins, we'd be, well, nowhere. Lots of different important protein molecules in your body. So how does DNA control the manufacture of the protein molecules that you're made of? Let's look at how it works with kind of a simple demonstration right here. So here we have DNA in the nucleus of the cell. So I've drawn a circle around it to represent that this is DNA in the nucleus. So that means the rest of the chalkboard out here, this is all the cytoplasm, all the stuff outside the nucleus of the cell. And way out here, I've highlighted, I don't know, can you see that dot? I hope so. There's one little ribosome, but it's pretty small, well, compared to the nucleus and everything else in the cell. So I'm going to enlarge it just so we can see close up what it's doing. So let me do this. Let me enlarge this ribosome and redraw it like there. Okay. So there's our ribosome enlarged. Now, let's look at the whole process of protein synthesis from beginning to the end. Now, there are instructions in this DNA for how to build a particular protein molecule. If you'll go back to the central dogma diagram, DNA makes RNA. We're going to look at how DNA makes, specifically in this case, messenger RNA. Here's how it works. The DNA unzips. It's all happening inside the nucleus of the cell. Now, initially, it kind of looks a little bit like DNA replication, but remember, we're not doing DNA replication right now. We're going to watch this side, the left-hand strand here of this DNA molecule. It's called the sense strand. We're going to watch it transmit its message into another molecule called messenger RNA. Another way you could put that is, we're going to watch this DNA molecule right here, at least this half of the DNA, we're going to watch it make messenger RNA. It doesn't really make it, but it serves as a blueprint or a template so that messenger RNA can be manufactured alongside of this left-hand strand, this sense strand of DNA. What I'm going to do now is I'm going to take the right-hand strand of this DNA molecule out of the picture. It doesn't really disappear like I'm going to show you here. It sort of hangs out patiently while its partner over here produces messenger RNA. So let me just get rid of this right-hand strand of the DNA molecule to avoid confusion over here. Now we got different kinds of enzymes involved here. There was one enzyme that was responsible, well, several enzymes responsible for replicating DNA. Now we've got a whole nother set of enzymes here responsible for taking this DNA and making messenger RNA. And then we've also got in the nucleus here, all of these, I'm going to call them RNA nucleotides. And they're going to be the building blocks that the enzymes are going to use to manufacture messenger RNA alongside of this left-hand strand of the DNA. Now you're going to see how, in a sense, the DNA, the order of the bases in the DNA, controls what the order of the bases in the messenger RNA will end up being because of the base pairing rules. A pairs with T, C pairs with G. Now one thing you're going to notice here is that RNA is a close cousin of DNA. It's a different kind of molecule. It's different. In fact, take a look at this picture of an RNA molecule that I've drawn. Now look carefully at it. There are three important differences I want us to notice about that RNA molecule. Can you spot at least a couple differences when you look at this picture of RNA? Well, you might notice that unlike DNA, RNA is single-stranded. You see that? It's just a single strand. It's like half a ladder. You wouldn't want to put an extension ladder like that on your roof and climb up on it. It wouldn't be safe. It's only half a ladder. RNA is single-stranded, unlike DNA, which is double-stranded. Now there's another difference if you look closely. Remember the four bases in DNA? A, T, C, G. What do you notice when you look at this RNA molecule? It doesn't have T. It doesn't have the base thymine or T. Instead, RNA has a very similar molecule. It's not exactly the same, but it's very similar in size and shape, called uracil or U. And that's another important difference between the RNA that you're looking at and DNA. Now there's a third difference that you really can't see in this picture, I'm sure, and that is that the sugar molecule in RNA, the one labeled S, it's not deoxyribose like in DNA, it's ribose. And so that's why this molecule's full name is ribonucleic acid, but we normally just say RNA because it's a whole lot easier to say. All right, now let's come back and look at the chalkboard. Let's watch messenger RNA being manufactured alongside the DNA. Looks like this A is going to be attracted to that T, And remember, there is no T in RNA, there's U instead. So this U will be attracted here. Remember, enzymes are controlling all of this. My hands would be like the enzymes. This G is probably going to be attracted to the C to the G, the U to the A. You think T, but remember there's R, there's U in RNA and not T. And then this G attracted to this C. Okay, we have just witnessed the process of messenger RNA being produced alongside this sense strand of DNA over here. And that process is called transcription. When DNA makes messenger RNA, that's called transcription. And so sometimes this mRNA molecule will be referred to as an mRNA molecule, and sometimes it will be referred to as a transcript. A lot of biologists today will use the word transcript instead. Now let's look at what happens next. The mRNA is going to peel away from the DNA that produced it, like this. Okay. And then the next step, the messenger RNA is going to leave the nucleus of the cell and venture out into the cytoplasm of the cell. And it can do that because unlike DNA, messenger RNA is single-stranded. So it can squeeze through the pores in the nuclear membrane right here. It can squeeze through, it can get through. So let's look at mRNA actually leaving the nucleus of the cell and coming out here into the cytoplasm. There it is. Now, you know what happens next? It attaches to a ribosome. So let's show that. All right, I'm going to just start reassemble it right here. So here are two of our different kinds of RNA molecules starting to work here. We've got mRNA. We've got rRNA, which is what the ribosome is made of. The ribosome is like this little wad of ribosomal RNA. And what's going to happen here is this ribosome, I'm going to do this just for the heck of it. This ribosome is going to read the messenger RNA. Now, the order of the bases in the messenger RNA spells out coded instructions for the ribosome on what amino acids to put together in what order. And the order is very important. Super important. Watch carefully. See this protein? If I do this, just switch these around. That's a totally different protein molecule. Watch this. And that's another totally different protein molecule. So the order of the amino acids is critical. You mess up the order of the amino acids and the protein won't work right. So that's very, very important. The ribosome is going to read these. That's what we say. It doesn't really have eyes like I'm showing you in my cartoon version here, but we say that it reads the messenger RNA. Now, there's a third kind of RNA at work here, and it's called transfer RNA. Transfer RNAs, they work a little bit like little taxis in the cytoplasm of the cell. And with my simple molecule here, the simple models, I've got a transfer RNA molecule right here. Here it is. Like all good RNA molecules, it's made of A's and U's and C's and G's. And this particular one has three very important bases sort of sticking out, kind of like landing gear. U, A, C. Sort of like landing gear. And this tRNA, like I said, is a little bit like a taxi or a shuttle. And its cargo that it carries is this particular transfer RNA's cargo is this molecule right here called methionine, which is one of 20 different kinds of amino acids that cells use when they're manufacturing protein molecules. So I'm going to attach this methionine amino acid to this transfer RNA. Transfer RNA molecules are floating around in the cytoplasm of the cell. All they do all day long is shuttle and deliver amino acid molecules to ribosomes. What pulls this transfer RNA molecule to the ribosome? I think if you look closely, you're probably going to see it. The attraction between the U and the A and the U, the C and the G. That's what's going to kind of propel or pull the transfer RNA to the ribosome. But it's all under the aid of special enzymes. Transfer RNAs don't just move around all by themselves. They have special enzymes in the cell that aid them in this whole process. There are a couple more vocabulary words that we need to look at. Every group of 3 bases on this messenger RNA molecule, for example, A, U, G. Each group of 3 bases is called a codon, because it codes for a particular amino acid. Take a look at this chart, and you've got a copy of this in your note-taking guide. Take a look at this chart. It's called the genetic code chart, and it shows all the 20 different kinds of amino acids in alphabetical order. Amino acid molecules with big, long, fancy names like alanine and asparagine and valine and methionine. So you've got all the 20 amino acid molecules listed on the left-hand side of the chart, and on the right-hand side, all the different kinds of mRNA codons. Can you find in that chart the codon AUG? I'll give you a second to find that. I'll give you a hint. It's just a little bit over halfway down the chart. AUG. Okay, according to that chart, what amino acid does the codon AUG code for? You are right, methionine. I hope you said methionine. That tells the ribosome that methionine is going to be the first amino acid in this long chain of amino acids that the ribosome is going to build, this long chain of amino acids that's going to be called a protein. So let's go back to the chalkboard here. Here, AUG. And sure enough, here's a special transfer RNA molecule carrying the amino acid methionine. It's going to deliver it to the ribosome like this. See how U is attracted to A attracted to U, C attracted to G? I already told you that this group of 3 bases on the messenger RNA molecule here is called a codon. You know what we call the 3 bases, like landing gear on the transfer RNA? You know what we call those that are the opposite or complementary to the codon? Those 3 bases on the tRNA, that's called an anticodon. That's what that's called. All right, so what's going to happen next? Well, look up CUG, the codon CUG in that code chart again. So here's the code chart again. So what amino acid does the codon CUG code for? I hope you said leucine. Leucine. So let's go back to the drawing board here. And lo and behold, what do you know? There's another transfer RNA molecule floating around in the cytoplasm of the cell, carrying its cargo, this amino acid right here called leucine. There it is. Check out the anticodon on the tRNA, GAC. Looks like it's going to be attracted to this codon CUG. Sure enough, here we go. It hooks up Looks like a spacecraft docking to the space station, kind of. Ka-chunk. In the meantime, the first tRNA leaves, floats back out into the cytoplasm of the cell in search of another amino acid to deliver to another ribosome somewhere. Then this second tRNA is going to unhook, float away. In the meantime, special enzymes are going to bond together, join together these two amino acids, kind of like that. It's going to bond them together, and all at once here, you can see that the ribosome really is more like a workbench, you might say. It's the place where all these molecules are working together to build a protein. Now, the protein molecule that we're building so far, you can see it's only 2 amino acids long. We're just starting. The messenger RNA in real life would keep on going, one codon after another, one amino acid after another being linked to this growing chain. And again, this is such a simplified model, but it gives us an idea, I think, because a lot of protein molecules are hundreds of amino acids long. Some are thousands of amino acids long. Some are hundreds of thousands of amino acids long. Some proteins are really big. But all these protein molecules that make up your body, they make up you, they run you in complex and wonderful ways.